Hello, I am Raji Banerji, a postdoctoral trainee at University of Colorado, Denver, and the first author of a recent publication in Brain Communications entitled as Enhancing Glucose Metabolism via Gluconeogenesis is Therapeutic in a Zebrafish Model of Dravet Syndrome. In this recent publication, we focused on correcting metabolic defects in a catastrophic pediatric epilepsy known as Dravet syndrome, which is caused by mutations in sodium channel gene SCN1A. The childhood disease is associated with frequent severe seizures, developmental delays, and SUDEP. Development of novel therapeutics for this field is crucial and necessary since most genetic epilepsies are resistant to FDA-approved anti-seizure drugs. The preclinical larval zebrafish model, referred as SCN1LAB mutant, has been validated by Dr. Scott Barabin from University of California, San Francisco. This mutant model is a valuable translational tool as it recapitulates key phenotypes of Dravet syndrome patients, such as spontaneous, unprovoked, and recurrent seizures and abnormal electrographic events. A major research gap exists in the understanding of metabolic dysfunction and identifying novel metabolic targets in genetic epilepsies. The most important clinical evidence is that Dravet syndrome patients respond positively to ketogenic diet, which is based on providing alternative mitochondrial fuels, suggesting that metabolic functions can impact neuronal excitability. Taking all factors into consideration, two questions arise. First, can we target metabolism to improve outcomes associated with the larval mutants? Second, will this uncover a novel and unifying metabolic mechanism that could contribute to Dravet syndrome disease pathophysiology? We developed a novel platform to study metabolism in these zebrafish mutants. Interestingly, we had previously reported reduced glycolytic and mitochondrial respiration rates in the mutants, accompanied by downregulation of key gluconeogenic genes, phosphoenol pyruvate, PCK1 and 2. In this publication, we validated hypometabolism and hypoglycemia as characteristic features of larval mutants. So could upregulation of glucose via gluconeogenesis correct hypoglycemia, metabolic defects, and consequent seizures in Dravet syndrome larval mutants? We adopted a strategy to pharmacologically upregulate gluconeogenesis pathway by using a metabolism-based small library screen. We identified a promising metabolic drug named PK11195, a well-known synthetic translocator protein ligand located at the mitochondrial outer membrane. This drug showed promising therapeutic potential as it activated gluconeogenesis pathway, reduced seizure-like swim behavior, and improved metabolic and glucose defects in the SCN1LAB mutants. The TSPO ligand was then tested for anti-seizure activity by electrophysiology in the mutants. Remarkably, a significant reduction in the frequency of ictal and interictal-like electrographic seizure events were noted with PK11195 treatment, thus confirming the anti-epileptic property of this drug. Here, I summarize the key findings of our manuscript. First, hypometabolism and hypoglycemia features were validated for the zebrafish model of Dravet syndrome. Most importantly, we uncovered therapeutic potential of a mitochondrial translocator protein ligand. This drug, PK11195, being a gluconeogenesis modulator, may serve as a glucose sensor and normalize impaired glucose metabolism, providing a novel therapeutic strategy to attenuate seizure activity in a preclinical model of Dravet syndrome. The highlight of this study is that these significant metabolism-based findings suggest that targeting energy-producing pathways is a viable and a novel therapeutic avenue for the treatment of Dravet syndrome and other genetic epilepsies. This study was conducted in collaboration with Dr. Scott Barabin of UCSF and funded by NINDS and Dravet Syndrome Foundation grants. Thank you for your kind attention.